Hey everybody, in this video we are going to look at transcription. So transcription, that is the process by which DNA is used as the template to make RNA. And it requires RNA polymerase. Now, prokaryotes typically have one RNA polymerase, and we will refer to that as RNAP. Eukaryotes typically have 3 RNA polymerases, and they can have more than that, but we will focus on 3, and they are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Now, RNA polymerase 1 in eukaryotes, it is in charge of transcribing most of what we call ribosomal RNAs. So rRNAs. Now, these are RNAs that fold into a certain structure and help form the ribosomes. RNA polymerase 2 is mostly in charge of messenger RNAs. Now, these are the RNAs that encode proteins. So these are the RNAs that are read by the ribosomes to make proteins. RNA polymerase 3, this one is mostly in charge of transfer RNAs. So transcribes genes that encode transfer RNAs. So transfer RNAs are used by during mRNA translation to synthesize proteins. We'll talk more about ribosomal RNAs and transfer RNAs in future lectures. We'll mostly focus on messenger RNAs. So you may hear terms like non-coding RNAs and coding RNAs. So there are many types of RNAs. The 2 examples of non-coding RNAs are ribosomal RNAs and transfer RNAs. So a non-coding RNA is essentially any RNA that does not code for a protein. So coding RNA, essentially only messenger RNAs are coding RNAs. Excuse me. So let's take a quick look at the structure of a prokaryotic RNA polymerase and a eukaryotic RNA polymerase. We don't need to remember too many details, but there will be one important, especially one particularly important part of RNA polymerase in bacteria that we will refer to, that I will refer to a little further on in the video. So with RNA polymerase, and we'll mostly focus on RNAP in E. coli. So it transcribes all RNAs. I forgot to mention that. So there's only one RNA polymerase. It transcribes all the RNAs, coding and non-coding in prokaryotes or E. coli. So structure is going to look something like this. I'm going to put in 6 subunits here. So I like to diagram it like this. Now, RNAP is a holoenzyme. And a holoenzyme, what's that? That's an enzyme with multiple subunits. The subunits are typically just proteins. Now, this is the omega subunit. This is alpha, another alpha subunit, so 2 identical subunits. This one is beta prime, and this one is beta. And then down here, we will have a sigma factor. So 6 subunits, 1, 2, 3, 4, 5, 6. and two of those subunits are identical. So alpha, omega, beta prime, beta, and sigma. So for RNA pol 1, 2, sorry, messed up there, and 3, now we already went over the different types, the major types of RNAs transcribed by the different polymerases here. Now we can say they all have a similar structure. With more than 10 subunits, and some of the subunits are shared between the different types of polymerases. So with respect to structure of these RNA polymerases, we just need to remember that it's slightly more complicated than the structure of RNA polymerase in E. coli. Okay, so now that we've covered that, let's take a look at gene structure, because RNA polymerase is going to transcribe a gene, and we need to know a little bit about gene structure before we talk about that. So some important concepts with respect to gene structure. Let's say this is part of a chromosome. It could be in a prokaryote, could be in a eukaryote. So a gene should have a +1 site, +1. Now the +1 site, we can also call that a TSS site, and that means transcriptional start site. So that is where RNA polymerase will start transcribing DNA, making an RNA molecule. Now down here, let's diagram I'm going to call this the TCS for transcriptional cleavage site. And that will be the point at which what we'll call the nascent RNA. So a nascent RNA is any RNA molecule that is still undergoing synthesis by RNA polymerase. So it's still attached to the RNA polymerase. And when you cleave the nascent RNA from the RNA polymerase to release the transcript, when the cell does that, not us, right? When the cell does that, it's no longer a nascent RNA. It's either a pre-RNA or a ribosomal RNA or a transfer RNA, no longer a nascent RNA. But as it's being synthesized, it's called the nascent RNA. In the transcriptional cleavage site, that's the site at which the nascent RNA is cleaved from the RNA molecule as part of becoming a fully functional RNA transcript. Now the part between the TSS and the TCS, this is the transcribed region of the gene. And when we talk more about protein coding gene structure in a future lecture, we will learn that students like to think that, okay, this transcriptional start site, this is also where the start codon for translation is, and that's not true. That's not true for a protein coding gene. For a messenger RNA molecule, you'll have the transcriptional start site up here, and then the translational start site, the AUG start codon, will be somewhere down here. And the same thing, the stop codon for translation will be somewhere before the transcript cleavage site. We'll talk more about that in later videos. So with respect to gene structure, we want to know, well, let me also address down here. Down here, we'll call this the terminator region. So down here, the terminator region of the gene. And this is where RNA polymerase will be dissociated from the template. Up here, and the promoter can sometimes overlap with the +1 site, and it can extend very far very far, but let's just say 1,000 base pairs. Let's say 2,000 to 1,000. It's hard to know precisely where a promoter region begins and where it ends. But the promoter region is at the beginning of the gene, let's say overlapping a little bit with the +1. And what it does, well, this is where RNA polymerase binds. RNAP, or the eukaryotic RNA polymerases, RNAP binds to the promoter. So it's the part of the gene that RNA binds to. And RNA polymerase binds to, and it needs to bind to the gene in order to transcribe it into an RNA molecule. And in order for RNA polymerase to bind to the promoters have what we call regulatory elements. And sometimes you might hear cis-acting elements. And this is sort of an old term. You almost never use it to, when I'm talking to my graduate students in the lab and doing research and discovering things, we almost never use this term cis-acting. So I'm going to call it a regulatory. We're going to call the things inside the promoter that, RNA polymerase binds to, we're called these regulatory elements. And these are DNA sequences. So there are certain DNA sequences that certain proteins, like RNA polymerase, or certain subunits of RNA polymerase, will bind, or when we talk about RNA polymerase binding to eukaryotic promoters, there are things called general transcription factors that bind to the regulatory elements. And they bind them because they have certain sequences that are recognized by these DNA binding proteins. So let's take a look at some regulatory elements so you can get an idea of what these things look like. So let me take a sip of coffee here. I have a little bit of a caffeine addiction, as I bet most of us do. So promoters have regulatory elements. These regulatory elements are important for getting the gene transcribed. So let's talk about two of the regulatory elements in prokaryotes. So let's say this is the beginning of a gene, and here's the +1 site. Now, if you were to look at, say, all the genes in E. coli, and look at all the +1 sites, and count back 10 positions, you would get what is called the -10 site. So the -10, that number comes from the +1. Wherever the +1 site is, the +1 being the first base pair that is, transcribed to make the RNA chain, count 10 bases back from there, and you're going to find, often you will find, not every time, but often you will find what is known as the PRIB now box. And so similarly, take the +1 and count back 35 positions to the -35 region. Again, this -35, that's the number of nucleotides from the +1 site. And if we said +35, that would be 35 nucleotides to the right of the +1 site. But -35, 35 nucleotides before the +1, we will often find what is called the -35 element. And it's not always there, because there can be other different types of regulatory elements in there that are in charge of, helping RNA polymerase bind. But in prokaryotes, often it's the PRIB now box at -10, and at -35, it's the -35 element. Now, what are these? So these are certain DNA sequences. For example, if we're looking at the -10 site in lots of genes from E. coli, we might see around that nucleus, around this region, around that -10 site, we might see something like this. A sequence like that. I'm not showing both strands of the DNA molecule. You can infer what the complementary strand is using the base pairing rules in the anti-parallel nature, right? of a DNA molecule. So I'm only showing one sequence, and this could be the -10 right here, or maybe that is actually this -10 here, but around the -10 region you might see something like that. And we could look at another gene and we might see a sequence like this at the around the -10 position, and we can look at another one and we might see something like this, and we can look at another one and we might see something like this. And we could look at hundreds of genes, do the same thing. Is every time we look at the -10 site, we see, wow, we see all these like T's and A's, and if we line them up, we might see a pattern. So at this position, we tend to see T. At this one, we tend to see an A. 1, 2. This one, we tend to see T. This one, we see 2 A's. If we look at more, you might see a T once in a while, but overall, the A's. And here we see a T. So this is called a consensus sequence. It represents the most common base at each position. So the consensus sequence. Consensus sequence. And consensus sequences are very common in molecular genetics. And they're important because they're used to describe the most common base at each position for related sequences, right? And if you have related sequences and related in a way that they have a similar function, it's very likely that they are evolutionarily related, because otherwise, if you compare random sequences, you wouldn't see a consensus sequence. You would see just random bases. And these tend to have the same base at each position because, they're related, and they're functional. They have a functional purpose. So they're less likely to mutate, because if you mutate or develop mutations and you eliminate this sequence, the protein can no longer recognize it. So here's the consensus sequence, and what this means is that there's probably some element that recognizes TATAAT, something that binds the promoter and recognizes this particular sequence of bases. And if we should change one of these, if this one up here, which isn't quite this guy right here, what it probably means is whatever typically recognizes this sequence, doesn't quite recognize this that well. It just binds it maybe a little less efficiently than it would say bind this one. We would need to determine that experimentally. But just because it's a little different doesn't mean that protein won't bind it. It just might bind it a little less well, and that might be good. That might be good. It might be a regulatory element. Well, the PRIB box is a regulatory element, so whatever recognizes it may bind it less well, and maybe RNA polymerase won't bind that promoter so well, and you'll get less transcription of a gene, say, with this sequence at the PRIB box than, say, the consensus sequence. Ideally, the consensus sequence is the one that is best recognized by the protein. And so the -35 element has a different consensus sequence. And in eukaryotes, we'll just look at one consensus sequence. Let's call this the +1. And if you count back 30 positions, so looking at a lot of different genes in, say, a eukaryotic species, if you find the +1 sites and you count back 30, you will often find what is known as the Tata box. And the Tata box has a consensus sequence, and there are proteins, and these are going to be the general transcription factors, and I'm going to talk more about those later, but they will recognize the Tata box, and they will be in charge of helping bring RNA polymerase to eukaryotic promoters of eukaryotic genes. And there are lots of other regulatory elements in here. And the point here is that I just want you to understand at this point what a consensus sequence is, how it doesn't have to be identical, but it essentially tells you the most common base at each position for related sequences, and understand what that genes and promoters of genes have different regulatory elements. And here are just 3 different types of examples, and the regulatory elements are defined by their consensus sequences. I think that's it for that. And know that the consensus sequences in DNA exist because they're bound by certain proteins. Okay, so let's go back to RNA-P. So the RNA polymerase in E. coli. Now we talked about the structure, and there was that sigma factor. So sigma factor, it turns out there are, I think, seven different sigma factors in E. coli. So 7 different genes that encode a sigma factor. So there are 7 different types, and we don't need to memorize these. But I think it's kind of interesting. They have interesting names. This one's a little different than these guys down here. RPOH, RPOS. Maybe the names aren't that interesting. But 7 different types, and we can take RPOE right here. So this sigma factor right here, it tends to be expressed when temperatures are high. So you move E. coli to an incubator. So you're growing a culture of E. coli, you move the culture to an incubator that is 10 degrees higher, then we should expect, we see higher expression of this gene right here, this sigma factor. Right here, if E. coli, the population of E. coli in the culture starts to exhaust the sugars in the solution and the media, and food is scarce, so this one is high. So expressed when food is scarce. Okay, so why would E. coli do that? And you know, I have this right here. This is from the notes that, you know, are available on ReggieNet. So 7 different factors, and each one is optimized for a different condition. So RPOD, which is sigma factor 70 here, optimized for standard growth conditions. This one optimized for high iron environments. This one optimized for movement, if it has to move. So in class, I believe I asked you guys, okay, to think about this. What would this do? Why would expressing RPOE, turning it on, making more of RPOE in a high temperature environment, why would that aid E. coli? And then I believe we looked up on Google how many genes are there in E. coli, and I think it was somewhere like 3,400 genes. So these are 3,400 genes, and every one of these genes isn't optimized for each of these different environmental conditions, right? Some of them would be needed more often under some environmental conditions than others. So it's most likely the case that, say, genes that are necessary at high temperatures, you might need genes that make decrease the fluidity of the plasma membrane, for example. We might need to turn those on when the temperatures are high. And they probably have regulatory elements in their promoters that are best recognized by RPOE. For example, so temperatures are high. A set of genes, there's a set of genes in E. coli that E. coli needs to turn on when temperatures are high, or needs to turn up when temperatures are high. Then they can do that by turning on RPOE, expressing that at higher levels. RPOE will then bind, will be incorporated into RNA polymerase, and RNA polymerase will be recruited to all that set of gene that is optimized to help E. coli survive that high temperature environment. So by turning on RPOE, RPOE is incorporated into RNA polymerase. RNA polymerase is then recruited to the promoters of genes that are required to help E. coli adapt to that high temperature environment. And you can think the same thing probably happens for all of these. So iron is too high. Okay, so somehow, and we have to go back a step to figure out what causes E. coli to turn on FEC-I, make more of the FEC-I sigma factor when iron is high. But once it does that, well, then it could be incorporated into RNA polymerase. RNA polymerase is recruited to the genes needed for E. coli to survive when the levels of iron in the environment are high. And there's going to be a set of genes, maybe 10 genes, maybe 100 genes, maybe 1,000 genes that the organism needs, is better suited to survive high iron environments when those genes are on. Okay, so essentially sigma factors, the different sigma factors, all in charge of helping turn on sets of genes that help E. coli survive better in different environmental conditions. So that's one way RNA polymerase in prokaryotes is recruited to certain genes under certain environments. With eukaryotes, we are going to divide recruitment of the RNA polymerases to eukaryotic promoters in two ways. Okay, so we are going to have basic recruitment, which we'll talk about now, and we will save advanced recruitment for later. So basic recruitment, now this involves the general transcription factors. Now each RNA polymerase has a set of general transcription factors that are in charge of recruiting that RNA polymerase to the promoter. And they have names like TF2A, TF, I guess I'm not sure if there's a TF3D, but they have names like this. And there are more unique names too. But there are a lot of these, and if you want to learn more about how these work, well, you definitely want to take BSc 350, which is molecular biology, where we talk maybe possibly too much about the mechanism of RNA recruitment to promoters, but I think it's fascinating. So hopefully I'll see you in BSc 350 if you want to learn more about those mechanisms. But okay, so the general transcription factors, what they do is they bind to promoters, the regulatory elements in the promoters, like the Tata box, and RNA polymerase, the different RNA polymerases, 1, 2, or 3. So they bring RNA polymerase to the promoters for transcription. So let me diagram this. Here's the +1 site. Let's say this is the Tata box here. And then here are some general transcription factors, and they are bringing in RNA polymerase here. GTFs. Okay. So advanced recruitment, what is that? Okay, so if you hear terms like enhancers, silencers, transcriptional activators, transcriptional repressors, and mediators. So these things are all involved with influencing the level of transcription. So general transcription factors, basic recruitment, this is necessary. All this is absolutely necessary just to get RNA polymerases on here and get transcription started. Then we have all these other things. Advanced recruitment is to really increase the level at which RNA polymerase is recruited to promoters to increase expression, or other things to silence or prevent expression. In this case, I'm using expression and transcription synonymously. But so advanced recruitment is going to involve all these complicated things right here to control the levels of transcription. Basic recruitment is just all the things that are necessary, just to essential. All this stuff right here is essential to transcribe the gene. Advanced recruitment are the things that are necessary for controlling the level of transcription. So for this part of the course, we are just focusing on how do you get RNA polymerase onto the promoter to transcribe the gene. And that involves expression of the general transcription factors, they will bind to RNA polymerase, and the promoter bring RNA polymerase to the gene to allow transcription to start. And we'll talk about these things later. Okay, so what do we need to know once RNA polymerase is there? What does it transcribe? Where does it transcribe? So let me diagram a DNA molecule. And there's a much prettier picture of this in the notes. So I encourage you to look at the provided lecture notes, which I think have a picture from the recommended textbook in them, or an image I grabbed somewhere from the internet. I'm not really sure, but I don't know if I can really show those in the YouTube video. So I will just diagram what I want you to know. Okay, so let's say that is the top strand of a DNA molecule. The top strand, or one strand of a DNA molecule. And I am diagramming the second strand here. I don't think I made any mistakes. It's possible. Now, this, let's say the plus one site is here. So this strand down here, I'm going to call this the template strand, and we're going to call this the non-template strand, which is also called the coding strand. Now, RNA polymerase is going to, recruit it to the promoter. In the case of prokaryotes, it was sigma factor. In the case of eukaryotes, it was the general transcription factors. RNA polymerase will be recruited. This will be unwound, and the mechanism by which that happens in eukaryotes is really fascinating. It involves one of the transcriptional factors. The way it's described is running along the double helix, and essentially all the other general transcription factors hold everything else in place, and by running along it, it kind of pops it open. That's really cool. But once it does that, it's unwound here, and RNA polymerase can start transcribing. So the first nucleotide that's put in is going, so this is the template strand. This is used as the template. So it's unwound, and then RNA polymerase starts transcribing by complementary base pairing. So we get a C, A, and then what's going to go here? Is going to be a T? Nope, it's going to be U, right? Because RNA uses U's instead of T's. So this U is going here, U here, C here, G here, A here, G here. So if I said, gave you a DNA molecule like this, I said this was the plus one site, I said this was the template, this was the non-template strand. I said, okay, give me the first, 10 nucleotides that are incorporated into the RNA chain. Well, you would say C, A, U, U, C, G, A, G. Okay, and you could continue that all the way down. Yep, U, U, A. And so what you might notice is that this strand right here is almost identical, right, to the non-template strand, which is why it's called the coding strand too. It's identical except for the T's are switched for U's. RNA polymerase is copying this one, but it's making a strand that is identical, making it a transcript that is identical to this one up here. Okay, and there's something neat about the process by, how does it get separated from this strand once it's being made? And let me diagram this a little bit for you. I may have some time here. So what would happen is we would, these strands have to be separated, right? You know what? No, I'm not going to do it. You have it in the notes, so you can see the pretty notes with the replication, the transcription bubble formed. An RNA polymerase is riding along the DNA template, and after about 7 or 8, I think, nucleotides, this nascent RNA is dissociated from the template. So you only have about a few nucleotides hydrogen bonded to the template at a time before it's dissociated. And the way it's dissociated has to do with the way it is sort of threaded through the RNA polymerase as RNA polymerase is walking around as a part of the protein that separates the hydrogen bonds between the RNA strand and the DNA template. Okay, so I think that's it for this introductory video on RNA transcription. We're going to talk more about RNA structure, and then we're moving on to translation next week in lecture. So see you next week. Hope you enjoy the rest of your weekend, and see you on Tuesday, or I think we have a study session on Monday. Okay, so see you in class or at study session or office hours. Okay.